So the innate is considered non-specific if we do the acquired immunity, which we're going to do in the next section. It's also called adaptive. Is there a C in there, acquired? And this is specific due to the antigen. Right, antigens involved. There are lots of antigens. And the definition of an antigen is just anything that elicits an immune response, and it elicits the acquired immune response, right? So non-specific innate doesn't, right? So the innate, you can group it into first line versus second line. And then we'll do in the next section the acquired. Adaptive immunity or acquired immunity has a memory as well, and it doesn't mean it's like, you know, got a brain or something, but it has, you generate memory cells, and that's just a term that was used, and those are white blood cells that they can respond within a couple days if you get re-exposed to an antigen. The first time you're exposed to an antigen, it takes about 2 weeks before you make an antibody, for instance. The second time, these memory cells allow you to make it within a couple days. So you have immunity once you've made memory cells. That could be a definition of it. You know, you could also say you're immune, meaning that you don't die from something. You know, you could say you're immune because you don't get real sick from it. So when you use the term immunity, it can have different meanings, unfortunately. And that's what makes it so difficult for people to understand, right? If you say children were immune to COVID-19, that was true, depending on your definition of immunity. If they didn't get very sick. Now, that doesn't mean every single child is immune, but you say children are immune, it's just, it wasn't incorrect to state that, in my opinion, whatever that is. So that's just the chart for that. So physical and chemical barriers are first line. By our skin, a mucous membrane, tears, even when we sneeze or cough. The acidity in our stomach is considered sort of a barrier. Most bacteria don't survive. You know, you take a probiotic, there's bacteria in it, most of them are going to die in your stomach. And then even, you know, urine flow helps cleanse. Ciliary defense would be first line. It, you know, helps expel things that are caught in the lungs. And then the second line, most of this section is about the second line defenses, and they're all innate, non-specific. If you have a white blood cell in your body eats a bacteria, it's non-specific. Now, it is, in a way, it's specific because for whatever reason, your normal flora aren't killed by your immune system for whatever reason, right? We don't attack our normal. So what our body does, right, the immune system is surveilling and looking for pathogens. But not only that, all of our cells in our body that are, you know, they're not on red blood cells, but we have this, it's called MHC class 1. There's a class 2. Somebody, you have what you refer to as a social security number associated with your MHC1. That's what it was. HLA is the other term used for this. So we discovered what MHC1, and people studied that, and then people were studying HLA, and then at some point we realized they were the same thing. Right? So there's two different names for the same. But I like to think of it as there's a social security number associated that is your tissue type. Another way of thinking of it. So you want to match, if you get someone else's tissue, you want to match that as closely as you can. And it's complicated, but there's hundreds of thousands of different numbers. Now, if your body, if you get someone's wrong social security number, or MHC1 or HLA, then your body will attack it and kill those cells. Okay, that's what. But you learn, your body learns it very early what's you. So the innate immunity, you know, and say other, you know, mammals all have acquired immunity. So the RE, or the old name was reticuloendothelial system, and it refers to a broad, it's kind of like your immune system, right? It refers to a lot of things in a lot of different parts of your body. This is the same thing. These are monocytes or mononuclear phagocytes. They're the things like dendritic cells, macrophages. They hang out in our tissue, right? They're not, say, like in our blood. There's lots of lymphocytes in what, you know, these, they call them lymphatic capillaries. They turn into lymphatic vesicles. And that whole system dumps into a spot in our bloodstream up here in the clavicle area, right? And that, when you have edema, that's what builds up, right? It's the fluid in between the cells. It's not in the red, you know, say in our bloodstream, right? And like this is the red vessel here, say the blood. And it, you know, it's really small at this point. And, you know, fluids or toxins that say build up in that fluid, if they can't get into the bloodstream to get cleansed, then they end up in your lymph system. It's called the lymph system because, or you know, you call them lymph vessels because there's lots of lymphocytes in there. And they're hanging out waiting for a pathogen. So if you have swollen glands, right, it's a swollen lymph node. A lymph node is just a, when these vessels come up, there's certain areas where they kind of get bigger, right? You call that a node. There's lots of white cells in there. So if you have an infection, they swell. So doctors, if they're examining you, they might check. You got some under your neck, I think, down here. They're all over the place. But you call that the lymphatic system, right? And here, it dumps right into the subclavian vein. You have like 1 million or 5 million red blood cells in a milliliter. You have about 10,000 white blood cells in the same amount. And then there are proteins. You find antibodies. Then when we get into section 14, you know, there are 5 different antibodies or antibody type. And right, what they do when they're produced, they stick to antigens, right? That's what antibodies do. Now this is, I'd like to just bring up this concept of maturation or activation. All right, if a cell is activated, it might turn into, it might mature a little bit, and then its function changes, right? That's, you could refer to that activation. When you make platelets, you start with a megakaryocyte, and that just fragments up. You know, erythroblast ends up turning into red blood cells. White blood cells, though, they have specific functions. But you can make all of them from one hematopoietic stem cell. That would be the most immature. Now, if you, say, take a monocyte and it moves into your tissue out of the bloodstream. It turns into a macrophage. It can also turn into a dendritic cell. So if you get a complete blood count, then the doctor is asking, you know, how many red blood cells you have per mil, how many white blood cells you have per mil. And if you get a differential with that, then he wants to know what percent are different types of white blood cells, right? So it might help him in his diagnosis. And hopefully you already know all of this, right? Erythrocytes are red blood cells. Leukocytes, white blood cells, right? There's lots of white blood cells, different types. So I'm just going to quickly review them. A neutrophil is a white blood cell that's involved in intracellular killing. And that just means that the bacteria, right, gets inside the white blood cell that kills it. And then it dies inside the neutrophil. There's also, macrophage is also used for this. And then we're going to go into how they chemotact and have to get to the area where the infection is, say, in your body. So we refer them as first responders. They're going to be the main white blood cell to eliminate, you know, say, bacteria that are somewhere in your body where they aren't supposed to be. Don't worry about the percents of all these. This is a gram stain, and these are all neutrophils, right? And they've gone to whatever area in your body you had this. This is actually a gonococcus, and because they're red, you know, pinkish or reddish, they're gram-negative diplococcus. This would indicate someone has a Neisseria gonorrhea infection. Just based on this gram stain, you'd be treated for that. So this is a nematode here, and all these, they have eosinophils, and it's attacking, right, all the eosinophils are attacking this nematode, and they'll kill it. They can't, you know, it's not like a bacteria, it's way too big, right? So in this instance, you call that extracellular killing. So that's what eosinophils do. They can kill things like red bugs, which are chiggers. So they, you know, it itches, a chigger itch, or any type of insect itch is probably from histamine being released by mast cells in your skin. You know, that's kind of how that works. But if you have allergies, then you normally have more eosinophils than someone who doesn't have allergies. And the basophil, you call it in your blood, but when it gets into your tissue, you say it's a mast cell. And then when we get into chapter or section 15, I'll explain how histamine is released by mast cells. And it's because you make an antibody to allergens that cause allergies. And they're found, those antibodies are actually found on mast cells. A monocyte, it becomes a macrophage. You call it a macrophage when it goes to the tissue. So those are white blood cells, macrophages and mast cells. They're there to surveil and get ready for, if you cut your skin, the mast cell will release, and I'll go through that in a minute, cytokines so that the neutrophils come in and attack whatever bacteria happen to get under your skin. And macrophages can phagocytize, but the neutrophils do most of it. So 20% of it, you don't have to worry about these numbers again, but right, natural killer cells are considered lymphocytes. And in the next section, we'll talk about how B and T cells, they're lymphocytes as well. But a natural killer cell will kill cancer, and they also kill cells that are infected with a virus. Now, when we talk about T cells in section 14, right? I've got them, you know, a T cell will also kill virally infected cells, but it's because those virally infected cells have an antigen from the pathogen. With natural killer cells attack cancer and viruses, but don't look for a foreign antigen or an antigen from a pathogen. They use other techniques. So those techniques are considered non-specific. Because T cells are specifically looking for an antigen, it's considered this, right, the acquired immunity. Right, killer cells belong under the innate because they're not looking for those antigens. You have an inflammation response because of an infection or an injury, and allergies is also under that category, right? An allergy really is just an immune response to something that's harmless, right? It's not, pollen is not going to hurt you, right? But your immune system, the problem with an antigen is they can be found on plants. So they can be in our diet. We can eat them. The same antigen can be found on an animal cell. They're just little, if you have your red blood cell, right, you have the A antigen on your red blood cell. It's a specific sugar. So this antigen or sugar can be found on things we eat. So that you have an A and you have a B antigen on red blood cells. Those antigens are ubiquitous. They're found in a lot of food things that we eat. We're constantly exposed to these antigens in our diet. So you refer to them as being naturally occurring. And we'll talk about that in section 15 some more. So when you have an inflammation, right, the area that's inflamed increases in temperature. It's not a fever. It gets warm. Then there can be reasons. It's increased your blood supply, but it goes up. It gets red. It swells. There's pain associated with it, so you know that you're injured if it's an injury. And then purpose, if it's an injury, you want to repair what's damaged, or you want to get rid of the pathogen or destroy the pathogen. Now this would be a scenario where your skin is, right, cut or punctured with something. Now they're showing a splinter here with bacteria on it, but even if you were cut with something that's sterilized, the normal flora that's found in your skin will get under the skin too. And it's not supposed to be there, right? It's only supposed to be on top of your skin. So regardless of what you get punctured with, it'll start this process. The mast cells and macrophages, right, they are tissue cells. That's where they're going to be there, and they're waiting for something like this to happen, right, where you get some a pathogen, a bacteria under the skin. They recognize it and then send out, dial 911. So you get a release of cytokines or chemical mediators, and there are 100 or more different interleukins. There's leukotrienes. So we just call them cytokines, or mediators is another word. But they're released and they draw in the neutrophils. So the neutrophils move through, chemotaxis, right, movement towards the chemical, which is released by the macrophage and mast cell. When they get there, then they begin to eat or phagocytize the bacteria. So they'll eat, 10 or 100 bacteria, however many they can, and then they die. So when you see pus is dead, most of it is dead neutrophils and dead bacteria. So if you give a cut, let's say, and you put Neosporin on it or something that contains an antibiotic, then it helps the process by killing the bacteria, and you can actually increase the time it takes to fully heal, the scab. You might lose a scab in, it might be twice as quick by using something like that. So chemotaxis is the movement of these, it can be any type of cell, right? But we're talking about phagocytosis. So in this case, it's white blood cells like neutrophils and monocytes. They are found in your bloodstream most of the time. They're only going to go to an area of infection, when you have an infection of some type, right? They're being signaled in by the mast cells. And that causes them to squeeze through, they squeeze between endothelial cells. That means they're going from the blood into the tissue, right? That's called diapedesis. Margination is just, they get close to the area where the infection is. And so histamine does all kinds of things, right? Increasing permeability makes it easier for them to squeeze between those endothelial cells, for instance. Like you have histamine receptors in your eye so that, the cells that produce tears, right, when you get watery eyes, well then they're responding to histamine by producing more tears in your eye. It could be a runny nose, right? The cells that make all the snot, they respond to histamine that way. So there's many responses. So mediators or cytokines, a cytokine is a chemical, kine means chemical, cyto means cell. It's probably the broadest term you can use. Like leukotrienes was used, right? Leuko referring to white blood cells. Triene means chemical in another language, I forget. So these are chemicals released by white blood cells, but there's many different leukotrienes, like there's many different interferons. Histamine is probably the most well-known one. It does all kinds of things, right? Permeability we talked about, vasodilation. Prostaglandins are released when cells are damaged automatically. There are enzymes that produce prostaglandins. There's a COX-1 and a COX-2. They're called the COX cyclooxygenase, but they produce prostaglandins. So if you take an aspirin, and aspirin inhibits this enzyme and prevents you or reduces how much prostaglandins you make, and that's why they cause pain. So it reduces pain that way. But the idea of this, mentioning all these are just to help you understand the concept of a mediator. Mediates the immune system or modulates, immune modulator. It means if you have an infection, you want it to work, turn it on, and after the infection's over, you need to turn it off. So if you're ever given, or if there's ever a need to use a steroid, the steroid puts your immune system to sleep. It modulates it down. So if you're dying from an immune reaction, then that might be appropriate, right? A steroid. But if you're trying to fight infection, the steroid's going to hurt that as well. So you kind of have to be careful. But the immune reaction can kill you before the pathogen does. That's what happened with a lot of people with COVID-19 in the early days. So prostaglandins, this is a list just to get, right, interferons, there are many of them, interleukins, those are another name. So phagocytosis is when white blood cells eat bacteria, and it's non-specific. It's kind of, you know, it's probably hard to explain why they leave normal flora alone. It's like they know they're good for us, so they don't destroy the good bacteria. All right, so if this is a bacteria, the bacteria have something on the surface. It's not an antigen, but just something, a component of their cell wall. And then there's a receptor on the phagocyte that sticks to that component. It can be different things for different bacteria. So a PAMP are found on bacteria or pathogens. I'm going to explain what that is in a minute. And then the PRR is the pattern recognition receptor that's found. This is the receptor here on the white blood cell. So the PRR binds to a component of the bacteria. That has to happen first. Now, if the bacteria has a capsule around it, that might block that interaction. So this explains how capsules make bacteria more pathogenic. We kind of already talked about that several times. So you adherence first, and then the bacteria gets inside. Intracellular killing, you have a vesicle, and inside the vesicle are bacteria. Right, this guy is now inside. Then you have a phagosome, it's called, for that. And then you have to digest it. So there's another lysosome that contains the enzymes that break down the bacteria. That's when you generate the phagolysosome. So you ingest something first, and then it's digested, is another way of thinking of it. Now, some bacteria, they can resist being digested. So PAMP, there are many of them. It stands for pathogen-associated molecular patterns. So pathogens are things like bacteria, in this case. So you could have a PRR on a white blood cell for phagocytosis. Now, in this depiction, the macrophage binds to a PAMP. We call it a toll-like receptor. There are different toll-like receptors that bind to different PAMPs. But let's say, for instance, it's double-stranded RNA. So the receptor here binds to double-stranded RNA, which is found in virus. When the receptor sticks to it, this is a PAMP, then this white blood cell dials 911 or releases things to wake up your immune system. So it modulates, it turns on your immune system when the toll-like receptors bind to pathogen-associated molecular patterns. So if you have immunity, say from COVID-19, right, And the first time you're exposed, you're making memory cells, but it's going to take, before you make antibodies to it, or T cells that can attack it, it's going to take 14 days before that can happen, if you haven't been exposed to it before. But the same day you start, say it starts making double-stranded RNA, then these cells, the innate immunity, can attack it. So this could explain partly why children were more likely to be okay, right? Maybe their innate immune system worked better against COVID-19 than ours. If you get Ebola, you have about a 50-50 chance of dying of Ebola. The only way you're going to survive it is if your innate immune system can take care of it and keep you alive for the first 3 or 4 days, 5 days. If you survive 10 or 14 days, then your antibodies are probably going to work pretty good against it at that point. So these are just some examples. The cell components of the cell wall, the lipopolysaccharide and gram-negative, bacterial DNA, and viral double-stranded DNA. So there's receptors that recognize these different parts of pathogens. And then if they do, it's considered to be part of your innate immune system. These are not considered antigens. Okay, they're patterns. So a pyrogen is something that causes fever. I'd like you to understand the term mostly. If you have bacteria that don't belong in your body again, or if they release an endotoxin, that can cause a fever, and viruses do too. The more viruses in your body, the higher your fever. It normally works that way. It probably depends on the virus too. Fever is considered a systemic response. It's not inflammation, right? It's your whole body. Your normal body temperature should be 37. Your hypothalamus regulates your body temperature. That's what I mean by that. What happens if you have an infection, the macrophage that eats the bacteria, say, and then it secretes interleukin-1 because of that. Interleukin-1 works at the hypothalamus to stimulate neurons, and they release prostaglandins that cause you to start to shiver, to increase your body temperature. So if you take aspirin, I had Cox up here. These enzymes produce prostaglandins. If you inhibit the enzyme with aspirin, then that reduces the amount of prostaglandins that are released and then reduces your fever. That's how an antipyrogen, they call it antipyretic activity, and whether it's Motrin or I think even naproxen, the NSAIDs, they're called, right? Non-steroidal anti-inflammatory drugs. Most of them that I know of are COX enzyme inhibitors. That's how they work. Right, so these are your non-narcotic. A narcotic binds to opiate receptors to reduce pain, right? And they can be addictive, whereas NSAIDs don't do that and are much safer. So what's the purpose of a fever? Right, just another list. You slow the growth of microorganisms, right? You get them out of their optimal temperature. They won't divide as rapidly. It also is a way of keeping the iron in your body from reaching the bacteria, right? It kind of limits that. They require iron. And then you also stimulate the white blood cells. So interferon, there's an alpha, beta, gamma version of it, and then there are alpha 1, alpha 2. Alpha 2 is the one that's used when you get treatment for, say, like hepatitis C or some type of viral infection. But if these were liver cells, hepatocytes, and this guy got infected with hepatitis C, it would release an interferon that would it would only affect neighboring cells. They don't release enough, right? It just meant to be a local type of behavior, right? So only nearby liver cells there would only be enough for them. They have receptors for interferon, and they would respond by making a antiviral protein to protect them from hepatitis C, hopefully. So that's a very simple explanation of how interferon works in that capacity. But like I say, there are, I think there's over 30 different interferons. You would consider this one at least, it's used to boost your immune system or protect you from pathogen. Might be a better way of putting it. But that's why it's used as a treatment for a viral infection. Now the complement proteins are considered innate or non-specific, but there's a caveat. Meaning one of the pathways, there's 3 of them altogether, right? We'll talk about the other 3 in a minute. There's the classical, which we're going to get into, alternative, and lectin. The 3 pathways are started with by 3 different mechanisms. The classical means antigen antibody, right? And that is part of the acquired immunity. At least the concept. You'd still consider all 3 of these non-specific, but it's a little gray area. Alternative is started with a endotoxin, and lectin is started with a sugar, by lectins. So how they begin, what sets them in motion, right? There's 3 different ways that can happen. So the classic pathway begins when an antibody sticks to an antigen on a cell. Let's just even say if this is a red blood cell, and you get an antibody attached to the red blood cell. There actually has to be 2 of them. There's a complement binding domain on these 2 antibodies. They have to be near one another, and they bind C1. And that begins the complement cascade. Now what ends up happening is the cell that these 2 antibodies stick to ends up being lysed or destroyed. Now if it's a bacteria, which it's meant for, then the bacteria dies. That's a good scenario. But if it's a red blood cell in your body, then it kills the red blood cell. So in section 15, a transfusion reaction, it starts with the complement cascade. That explains that. And we'll talk about it a little bit more in section 15. In this section, when you say you coat a bacteria Right, that's a red. Let's say we have a bacteria here, and then 2 antibodies are sticking to it. Even before you get the complement, when you coat this bacteria, it's called opsonization. It's coating a bacteria with antibody. Now, when you coat it with antibody, it can more easily attach to a neutrophil that wants to, phagocytize it. So you see, that's how it enhances phagocytosis. Now, the other thing that works is C3b. Let's say those are squares, right? You can coat bacteria with C3b. So these antibody and C3b are considered opsonins. And when they coat the bacteria, then you enhance phagocytosis with them. That's called opsonization. The reason I mention this is now we're going to get into the complement cascade and generate C3b. Okay. Now, like I said up there before, C3b is opsonization. C3a is involved in inflammation. The way those two are generated, first, to start the classical pathway, antibodies bind to a bacteria or a cell. Then C1 sticks to the antibodies. C1 activates C2 and C4. Then 2 components of them get together, activate C3, and then you literally cut C3 in half. Right? They think of it as you activate it is another way of describing it. Now, C3b can be used for opsonization, meaning go on and coat the bacteria that's the antibodies attached to, right? This guy up here. When you generate C3a, it's going to signal inflammation. So the area where the bacteria is becomes inflamed. Right? It's part of that process. C3b also continues the complement cascade. And then what you want to eventually do is punch a hole in the bacteria to lyse it. That you form what's called a membrane attack complex, which consists of C9, C8, C6, and C7. And I'm thinking that'd make a great test question, wouldn't it? What composes the membrane attack complex? Now, don't worry about the order or the numbers, right? You should remember C3b is an opsonin, C3a is inflammation, but you don't have to worry. I kind of want to go through the cascade so you understood it. It's kind of like the clotting cascade is similar. You learned that before. It's a series of proteins found in your blood, made by the liver. They all work together. The outcome of activating complement, or each number is a different protein. C1 is a protein. C2 is a protein. And they kind of put them in order, but not completely, unfortunately. They're not. So that just, the main purpose is to lyse the cell that the antibody is attached to. Now, if it happens to be a red blood cell, the red blood cell is lysed as well. But we still, even though you get an antibody and an antigen interaction, we still consider this not specific. Right? That's a minor point. Now, these two, alternative and lectin, don't require C1. Because, right, so they can start without antibodies. They can still get the membrane attack complex, but they're not initiated with antibodies. So they're definitely non-specific. Antimicrobial peptides, right, do exactly what they sound. They work against microbes. They can lyse cells or inhibit cell wall synthesis, so they can, you know, I don't really need, you know, I'm not naming any of them and expecting you to remember which each one of them does. Let's just keep it that simple.